Preparation Model Paper 20. I'm Anish Shankar Reddy, PhD Plant Pathology. First one is frog eye spot disease can be observed in. So they have mentioned the crops. In which crop we used to observe this frog eye spot. Generally, this frog eye spot is caused by Cercospora nicotiana, right? Potato, tomato, tobacco, and cotton. So generally, frog eye spot we used to study in the section of tobacco. Frog eye spot of tobacco is caused by Cercospora nicotiana. So the answer is tobacco. Mat-tailed sap rot of tamarind is caused by fungi, bacteria, virus, and viroid. Generally, this disease is caused by fungi. Black tip disease is associated with. So, in which crop we used to study this black tip of mango, banana, guava, mango, and tomato. Generally, this black tip of we used to study in the crop of mango. So, generally, this black tip of mango is caused by boron deficiency, and also one more reason like sulfur dioxide concentration or sulfur dioxide toxicity is also one of the responsible for this black tip of mango. Simply, if you see black color tips or black color spots can be observed in mango. Sometimes it may confuse with mango anthracnose, but the only difference with mango anthracnose and the black tip is mango anthracnose is initially small spots, later coalesce and look like a very big spots, but this black tip is comparatively anthracnose, the spots are little bit smaller. And the next one is rescue treatment fungicide. Which fungicide is generally considered as a rescue treatment fungicide? Tridamon, triadamifon, tridamorph, metaloxyl, mancozeb. Generally, for powdery mildew, we recommend tridamorph group of fungicide. For metaloxyl, oomycetes, mancozeb, we can recommend for almost all kinds of crops, right? So this triadamifon, the trade name is Bileton. So the trade name is Bileton is generally considered as a rescue treatment fungicide. Griffith performed the transformation experiment in mice, rat, rabbit, and cow. Majority of the experiment scientists used to conduct in mice only. The two white mice. The mice are very small. Even when we see the zoology people, all kinds of experiment are they used to conduct in this mice only, the two white mice, where the responses are fast and we can get the results as much the fast as possible. So the Griffith performed this transformation phenomenon in 1928. So this transformation is a kind of sexual recombination in bacteria that conducted in pneumonia, right? Pneumonia. And the next one is a marsh spot of pea is caused by. So it is generally a zinc deficiency. Marsh spot of pea, little leaf of rice Kara disease, white bud of maize. These are all caused by zinc deficiency only. The protective medium used in a cryopreservation or cryo storage for fungi. So for fungi, what is the different kind of cryopreservatives are used? 10% glycerol, dimethyl sulfoxide, glycerin. Actually, this glycerol, 10% glycerol and dimethyl sulfate, both A and B can be used in long-term preservation. For example, a couple of preservations we use dimethyl sulfoxide that can be stored up to 10 to 15 years, and some kind of preservation we use 10% glycerol that can be helpful in preservation for 10 years or maybe 15 years, it depends, but both glycerol and dimethyl sulfoxide can be recommended as cryopreservants. I think the same question was asked. The answer is both A and B. Glycerol and dimethyl sulfoxide can be recommended for cryopreservation or cryo storage. The first person to use formaldehyde for the preservation of potato scab. Who is the first person who used formaldehyde for the preservation of potato scab is J.C. Arthur. J.C. Arthur is the first person who used this formaldehyde. Whittaker is largely known for five kingdom classification, right? So then five kingdom he just separately erected fungi as a new kingdom of classification. Kirketal, the recent classification and most widely used classification that we are following, that is 2008. And the next question is the most commonly occurring plant bacterial diseases. Most commonly occurring spots and blights, leaf curls, wilts, and diebacs. Generally, mostly we can see spots and blights outside. Buller drop is associated with spore discharge, spore germination, spore penetration, all the above. Generally, this Buller drop is generally related to spore discharge. If you see the spores during a discharge or during maturation, there is a small bubble or water drop kind of structure can be observed on the base. So this creates enormous turgor pressure and pressure. So this also helps to create the pressure that will shoots. So this Buller drop is actually kind of a spore discharge mechanism, where we can observe in spore discharge mechanism. The first commercially defined synthetic media for nutritional studies of Aspergillus niger was developed by. So the first commercially synthetic media was first developed by J.K. Rowling, that I know very well. Robert Petrie, Rowling, and Anton de Barry. As I told you, Rowling was the first person who developed the synthetic medium for culturing of organisms. Whatever it is, the organism, you first remember the first synthetic medium or chemically defined synthetic medium was first developed by J. Rowling. Synascus or compound ascus is produced by Pythium root rot, wheat powdery mildew, ergot, sorghum, or coriander stem gall. So this coriander stem gall is the one more characteristic feature of Synascus or compound ascus. This coriander stem gall is caused by Protomyces macrosporus, where it is produces Synascus or compound ascus type. Wound dressing fungicide. Which of the following is a wound dressing fungicide? Bodo paste, Chaubetia paste, Bodo mixture, and both A and B. Generally, paste or paste, any paste, Bodo paste or Chaubetia paste, whatever it is, the paste kind of substances are generally recommended for wound dressing. So once we can observe wound on the plant surfaces, or once we have done after grafting or maybe some other kind of operations, if there is any kind of wounds can be observed, that can be treated by using, or that can be covered by using this kind of paste kind of substances. So the answer is Bodo paste and Chaubetia paste can be recommended for wound dressing fungicide. The bacterium whose cell wall contains cellulose. Generally, bacterial cell wall is composed of peptidoglycan or mucopeptides. There are a couple of bacteria whose cell wall contains cellulose also, like Histobacterium xylinum, Histobacter acetizinum, Zymosarsina ventriculi. So these are all the couple of bacteria that I have given, and so the answer is all the above. So all these bacteria contains cellulose in their cell walls. Carborundum powder is chemically known as ammonium sulfate, ammonium chloride, silicon carbide, copper sulfate. Carborundum powder is known as silicon carbide. Silicon carbide is known as carborundum powder chemically. So where it is used in the local issue, say. Horizontal and vertical resistance are the forms of, so non-host resistance, true resistance, apparent resistance, all the above. Generally, in the true resistance, where the true resistance is actually divided into two kinds of other resistance, that is a vertical and horizontal resistance, right? So the answer is true resistance. In true resistance only we can observe this vertical and horizontal resistance. The term symbiosis and heterosism, is coined by Anton de Barry, Micheli, Ward, Secado. Generally, this symbiosis and heterosism generally related to Anton de Barry. So while he was working, he termed the concept called symbiosis, where the mutual relationship, and heterosism in the while he was working with wheat stem rust, autoecious and heterocious nature of the wheat stem rust. That is also given by Anton de Barry. P.A. Micheli, founder and father of mycology, he wrote a book called Nova Plantarum Genera. H.M. Ward, father of tropical plant pathology. Secado, Psyllum fungorum, he wrote Psyllum fungorum, right? And the next one is the Shiffard's crook symptoms is associated with. So which of the following diseases is associated with Shiffard crook symptoms? Bacterial blight of rice, black arm of cotton, fire blight of apple and pear, and ring rot of potato. So this Shiffard's crook symptom is associated with the fire blight of apple and pear. If you see the infected twigs or stems at branches at the end, it is little bit curled like this. So due to this curling, this kind of curling is known as Shiffard crook. Such kind of symptoms we can observe in fire blight of apple and pear. So the next one is the type of secretion system present in almost all plant pathogenic bacteria. All plant pathogenic bacteria contains type 1 secretion system. So the type 1 secretion system is most common in and found in almost all plant pathogenic bacteria. A virulence gene was first isolated by who isolated the first this A virulent gene. So Van der Planck, Alexander Fleming, H.H. Flor and Van Loon. So generally this A virulence concept was first given and first isolated by H.H. Flor. So the term A virulent gene also coined by H.H. Flor in 1950. Van der Planck, we know very well, is considered the father of plant disease epidemiology. Alexander Fleming, world's first antibiotic, Penicillium notatum. Penicillin isolated from Penicillium notatum. Now it is isolating from Penicillium chrysogenum. Protein for protein hypothesis was given by protein for protein hypothesis was given by Van der Planck. So Van der Planck only gave protein for protein hypothesis. So Butler, father of plant pathology and mycology in India, E.J. Butler. E.F. Smith, father of plant bacteriology, and also the man who gave the fourth Koch postulates. Out of four Koch postulates, three was given by Robert Koch and the fourth was given by E.F. Smith. Okay, the sheath blight of rice was first reported in India from Punjab, Andhra Pradesh, Tamil Nadu, Kerala. The sheath blight of rice was first reported from Punjab. Annual review of phytopathology was published from which year? The annual review of phytopathology is publishing. It is publishing from the year of 1963. The next one is the dapple apple is caused by fungi, bacteria, virus and viroid. Dapple apple is the disease related to viroid. It is a viroid disease. And sun scorch of areca nut is caused by fungi, bacteria, abiotic disease and viroid. So generally the sun scorch is actually abiotic disease due to the name itself indicates a sun scorch. Due to heavy temperatures, the scorching can be observed. So this is abiotic disease. The first book completely devoted to biological controls, ecology of soil-borne pathogens, biological control of plant pathogens, biocontrol of plant pathogens. So generally there are two persons like Baker and Cook in 1983. They wrote a book called Biological Control of Plant Pathogens. That book is regarded as the first book that is completely devoted to biocontrols. So Baker and Cook wrote a book called Biocontrol of Plant Pathogens 1983. When we ask what is the first book that is completely devoted to plant pathology, diseases of cultivated crops and their causes and their control, which was written by J.G. Kuhn in 1858, right? So that book is considered as the first textbook that is completely devoted to plant pathology. First expert system developed against which diseases? Soybean, potato, tomato, grapes. So first expert system which was developed by USA in 1983, especially for 17 soybean diseases. So the crop is soybean. For 17 soybean diseases, this expert system was developed by USA. This expert system is considered to be the first expert system in 1983. First recognized viral diseases in the world: tomato mosaic virus, cucumber mosaic virus, yellow mosaic virus. The first recognized viral disease in the world is the tobacco mosaic virus that was first discovered by Bijernek in 1898, and is also the one who coined the term virus, right? So give an example of chlamydospores producing fungi. So which of the following fungi is a chlamydospores producing fungi? So Verticillium, Fusarium, Phytophthora, alternaria. So actually, generally out of the given options, this Fusarium produces a chlamydospore as one of the spore. So Phytophthora produces sporangia, alternaria conidia, like bell kind of shaped conidia with vertical and horizontal substations can be observed, right? So this Verticillium only typically produces this chlamydospore. So microconidia, macroconidia, and chlamydospores are produced by this Fusarium. Chlamydospores are generally produced during unfavorable situations, right? So the answer is a Fusarium. Groundnut bud necrosis is transmitted by thrips, whiteflies, eriophyid aphids. Groundnut bud necrosis virus is generally transmitted by thrips tabaci. Water as a medium of preservation of fungal cultures was first suggested by, which means in water also we can preserve fungi, were first suggested by Robert Koch, Hartig, uh, uh, Castellani and Anton de Bary. So this Castellani was the first person who suggested that this fungi can be preserved in water by adding other things also. So, but the first person who given by Castellani. Okay. Teleomorph of trichoderma. Teleomorph means sexual stage of trichoderma. Diplocarpon, Hypocrea, Glomerella, Fulvia. Hypocrea rufa is generally the teleomorphic stage of this trichoderma. We know very well this trichoderma is the best biocontrol agents and the best fungal biocontrol agents that we are mass producing and using all over the India, right? Cutted disease of cardamom is transmitted by. So they're asking transmitted by. Okay. So if it's Pantalonia caladi, whiteflies, mites, mealybugs. So this cutted disease of cardamom is transmitted by black aphids. So Pantalonia nigronervosa and one more Pantalonia caladi. Not only this cutted disease of cardamom, banana bunch type is also transmitted by this aphids or black aphids, Pantalonia nigronervosa, right? The next one is a biocontrol agent used for the control of root rot nematode. So which of the following biocontrol agent is used for the management of root knot nematode? Okay, Pseudomonas fluorescens, Bacillus subtilis, Pasturia penetrans, Dactylella species. Generally this Pasturia penetrans, even in the biocontrol section also we have saw this Pasturia penetrans is the best recommended biocontrol agents against nematode disease, especially this root knot nematode is especially managed by using this Pasturia penetrans. Okay. Apothecium means, so there are four kinds of sexual fruiting bodies are there: Clistothecium, Apothecium, Perithecium, naked assay. Clistothecium means completely closed ascocarp. Apothecium means completely opened ascocarp. Perithecium means partially opened ascocarp. Naked assay means no ascocarp. Generally, naked assay can be observed in a peach leaf called Taphina deformans. Very important one, right? This they have asked, right? What they have asked is apothecium means completely closed clistothecium, completely opened apothecium, partially closed ascocarp or partially opened ascocarp also we can say. This partially closed ascocarp or partially opened ascocarp both are belongs to this perithecium, and what they have asked is apothecium, so it belongs to completely opened ascocarp. Like this it will be completely opened where the asca are produced naked outside, not naked outside. Okay. Who reported first phloem inhabiting bacterial diseases? So E.F. Smith, Robert Koch, Winston Black, H.M. Ward. So Winston Black was the first person who reported this fastidious vascular bacteria for the first time. Example of compound interest diseases: rust, powdery mildew, lead blight of potato. All are the examples of compound interest diseases. So simple interest is generally leaf blights or spots. Generally we can say. Simple interest diseases are also known as monocyclic diseases. Compound interest is also known as polycyclic diseases, right? Protein subunits of TMV consist of. So there are how many proteins are generally present in TMV? They're asking 158, 2130, 6400, and 168. Generally 158 amino acids, 2130 protein subunits, 6400 nucleotides are generally present. Since they have asked protein subunits, it's a 2130 protein subunits are present. Effector protein in plant pathogenic bacteria is released through the secretion system. So which secretion system is involved in the release of this effector proteins? Generally type 3 secretion system plays a very important role in this pathogenicity and the release of this effector protein in plant pathogenic bacteria. We know very well around 7 kinds of secretion systems are there: type 1, type 2, type 3, type 4. Type 3 type 1 is the majority of plant pathogenic bacteria, type 4 is agrobacterium, type 3 is like Xylella xanthoma, some kind of bacteria we can observe, right? So these effector proteins are generally produced by this type 3 secretion system. Okay. Redomil MZ is a fungicide consisting of metaloxyl, mancozeb zinc oxide, metaloxyl plus mancozeb combination. So this metaloxyl plus mancozeb combination they are selling outside with the Redomil MZ. So Redomil is the trade name of Redomil Gold is the trade name of combination fungicide like metaloxyl and mancozeb. Mancozeb and metaloxyl combination. So such kind of combination will the companies are selling outside with the trade name of Redomil MZ or Redomil Gold. first toxin to be isolated in a pure form. So which toxin is isolated in a pure form for the first time? So wildfire toxin is the toxin which is produced by Pseudomonas syringae, patho or tabasi, I think. That is the first toxin to be isolated in a pure form. Citrus greening is caused by, so Xanthomonas axonopodis pathovaricity, Candidase liberibacter asiaticus, Xanthomonas compressus pathovaricity, and Xanthomonas varice. So if you see Xanthomonas axonopodis pathovaricity which causes citrus canker, Candidase liberibacter asiaticus which causes citrus greening, Xanthomonas compressus pathovaricity, I have no idea about this. Xanthomonas varice, pathovore varice, which causes rice bacterial leaf blight. Since they have asked citrus greening, it goes to Candidatus liberibacter asiaticus. It is FEB, okay, Fasciolus vascular bacteria. Next one, little leaf of brinjal is transmitted by aphids, leafhoppers, mites, and whiteflies. First of all, we must understand what is this little leaf of brinjal. So it is a phytoplasmal disease, little leaf of brinjal, Sesame phyllodes, mulberry dwarf, apple proliferation, sandal spike, all these are little phytoplasmal disease. Majority of the phytoplasmal diseases are transmitted through leaf hoppers only. So if it is a phytoplasmal disease, majority of the phytoplasmal diseases are transmitted by leaf hopper. Of course, other vectors also be a part of it, but majority of the phytoplasmal diseases are transmitted by leaf hopper. So the answer for the given question is leaf hoppers. Banana bunch top was introduced in India from which country this banana bunch top was introduced, right? So Europe, USA, Sri Lanka, Netherlands. So it is believed that banana bunch top is introduced from Sri Lanka. And the oldest fungicide used in the control of plant diseases, sulfur, bodomixture, copper sulfate, jiram. It is well known that sulfur is used from centuries. So this is known to be the most oldest fungicide used in plant disease management. Next one is the plant origin resistant gene which encodes a detoxifying enzyme. So which gene that encodes a detoxifying enzyme? HM1, PTO, Xanthomonas 21, CF9. So this is HM1 and also known to be the world's first resistant gene HM1, Helminthosporium victoria. That is HV toxin. Sorry. This Helminthosporium maize. It is isolated from maize, right? So this is known to be the first resistant gene, and it actually encodes with the detoxifying enzyme. What is the specific antibiotic used for the management of phytoplasmal disease? So tetracycline is well known for the management of phytoplasmal diseases, and it is also known to be that sometimes the plants also can be recovered a little bit, but I was a little bit doubted about whether the plants can be recovered or not. But generally for the management we can recommend this tetracycline, that is well known. The primary inoculum of cucurbits wilt, that is striped and spotted cucumber beetles. So what is this primary inoculum? Midgut, foregut, body and abdomen. So generally on the body itself is where this striped and spotted cucumber beetle can transmit. So this body can be considered as a primary inoculum, like a whole body or the whole insect. Conidiophores of which of the fungus arises from the thick-walled food cell? Penicillium, Aspergillus, Plasmodiophora, both A and B. So generally this Penicillium and Aspergillus, where if you see their conidiophore arranged from thick-walled food cell, but inverted T-shaped food cell can be observed in Aspergillus, but inverted T-shaped food cell is present in Aspergillus, absent in Penicillium. Since the question they have asked is conidiophore arised from the fungi of thick-walled food cell. Generally both Penicillium and Aspergillus arise from the thick-walled food cell, but inverted T-shaped food cell is only observed in Aspergillus. Moniliformin and aflatoxins are mycotoxins, phytotoxins, pathotoxins and vivotoxins. So this moniliformin, which is produced by Fusarium kind of species, Fusarium moniliformi, aflatoxin, both are mycotoxins. Okay, so these phytotoxins, pathotoxins or vivotoxins, they're all kind of toxins which plays a role or in pathogenesis and the stuff. So but this moniliformin and aflatoxins are the parts of mycotoxins. The next one is minute projections near the point of attachment of sterigma to the basidiospores are known as promycelium, paraphysis, clamp connection, hilar appendix. So this hilar appendix were generally the minute projections near the point of attachment of sterigma. So the name of this attachment is known as hilar appendix. Rust of P is caused by, generally rust of P is caused by Uromyces fabae, right? So Puccinia pathway is associated with, they are asking associated with which disease? Wheat stem rust, wheat smut, linseed rust, all the above. So Puccinia pathway and wheat stem rust also both are associated with wheat stem rust, which is caused by Puccinia graminis etc. This Puccinia pathway was given by Nagarajan and Joshi in 1980. Wheat stem rust rules or Indian wheat stem rust rules was given by Nagarajan and Singh. Puccinia pathway was given by Nagarajan and Joshi in 1980. RNAi technology is used for, RNA means RNA interference. Okay, that was first given by Andrew Fire and Craig Mello, right? Sorry, Andrew Fire and Craig Mello only. Yes, I think so. The technology was first introduced and discovered in a nematode called Rhabdis elegans, and they sequenced and got Nobel Prize in later stages as well. This RNA interference technology is used to promote the expression of genes, suppresses the expression of genes, methylation of genes, and the report of genes. Generally, this RNA interference, it cuts the expression of the genes. So the answer is the suppression of expression of genes. Soil-borne pathogens can be best managed by, so fungicide application, hot water treatment, or biocontrol agents or mixed cropping. Generally biocontrol agents are best recommended or best known for the management of soil-borne pathogens. Induced systemic resistance is triggered by the following of Colostochum gliosporites, Pseudomonas fluorescens, Pseudomonas solanacearum, Bacillus thuringiensis. Generally Pseudomonas fluorescens has a ability to trigger this induced systemic resistance. So the answer is Pseudomonas fluorescens. Streptomyces cabis can be the best managed by application of antibiotics, soil pH management, flooding, and seed treatment. So Streptomyces is the best known for the management of soil pH management. For example, Streptomyces cabis, potato scab is severe at 8.0, around as I would say a saline pH, right? Saline. So more than base. So if you reduce bring down the soil pH to around 5.2 to 5.5, we can effectively manage this potato scab, which is caused by Streptomyces cabis. Generally, Streptomyces is an antibiotic, but in only 1 or 2 cases, especially in the case of potato, which causes a disease, that is a potato scab. So if you bring down the soil pH by the application of gypsum, around probably 900 kilograms per hectare can be recommended. So if you apply gypsum, we can bring down the soil pH. If you want to increase the soil pH, we can recommend lime. Okay, for the management of application of lime can be the best management practices for clubroot of cabbage. So if you increase the soil pH, we can manage that disease. In the case of potato scab, we need to reduce the soil pH from 8.0 to around 5.5 to 5.2 to 5.5 or close to 5.5 to 5.7 below. So managing the soil pH management only. So X disease of peach and plum is caused by Phytoplasma, Spiroplasma virus and viroid. So it is caused by Phytoplasma. X disease of peach and plum is caused by Phytoplasma. Which one of the following is different from the group? Grape vine yellows, potato spindle tuber viroid, citrus ixocotosis, coconut kadang kadang viroid. So the answer is potato spindle tuber, which is actually a different group from the given viroids. For the preparation of 5 liters of 0.5% Bodo mixture, the amount of copper fungicide required is very simple, 5 gram. Like there is a fungicide calculations are there. Now it will be, again it will take time to explain all the fungicide calculation, how it will be started. So for 5 liters, simply there for preparation of 5 liters of 0.5% solution, the Bodo mixture compound, the amount of copper sulfate required is 5 grams. Generally they are preparing for 5 liters, right? So you just think about it. The composition of body mixture is 1:1:100. 1 kilogram of copper sulfate, 1 kilogram of hydrated lime, 100 liters of water. For 100 liters, it is required for 1 kilogram. So if it is a 1 liter, we required 1 gram, right? So if it is a 5 liter, we required 5 gram. Very simple. So the next one is ITS is commonly used for the identification of. So ITS is generally commonly recommended for the identification of fungi, bacteria, virus and nematodes. Generally for fungal identification, it is the most commonly universal primers recommended for the identification of fungi. Dahlia mosaic virus is an example of. So their genome they are asking. Simply Dahlia mosaic virus contains a genome of double-stranded DNA. Murin is present in which bacteria? Which of the following bacteria the murin is present? Methanobacterium, cyanobacterium, halobacterium, and clavibacter. General clavibacter contains this murin. First international phylloxera convention held in the year in 1881. I believe the first international phylloxera convention happens. So this phylloxera is the main reason for grapes downy mildew. Okay, and it is also the most problematic, especially in grapes and other crops as well. So later scientists came to a conclusion that we must control this disease. So all gathered in a place and started a convention called phylloxera convention. And it is also known to be the first international convention, that is the phylloxera convention, that happened in 1881. The next one is total number of chromosomes are present in bacteria. Bacteria contains only one chromosome. Okay, and it is also the important one, repeatedly asked question. The red pigment producing bacteria. So which bacteria produces red pigment? Serratia, Staphylococcus, Micrococcus, Monococcus. So Serratia is known to produce as red pigment. It is also known to be the human pathogenic bacteria. Okay, Serratia marsan produces a red color pigment, especially in milk. If you see the milk, it is infected with Serratia, we can see the red color kind of pigments. Disease prediction stimulation model, APPRE, is related to. APPRE is actually related to serial rust disease. Okay, sex organs, that is spermatia and receptive hypa formed on smut, rust, downy mildew, powdery mildew. It's a special kind of reproduction only observed in rust only. Even in the sexual reproduction also, we used to study spermatization, where the spermatia and receptive hypa combined and where reproduction happens. So such kind of things we only observed in rust, wheat stem rust kind of things, right? Who proved dust as a carrier material of germs by conducting an experiment in a specially designed box. So Spallanzani, John Tyndall, Schwann, Anton Lewinck. Generally, this John Tyndall was the first person who proved that this dust act as a carrier material in transforming or spreading of the germs. Covered smut of barley is caused by the fungus. So covered smut of barley is caused by Stylagus segatum pharma species Hardy. So barley scientific name is Hardy vulgar, right? Like that. So downy mildew of soybean is caused by Pernospora manchurica. It is also an important pathogen in soybean. And the stem gall of coriander survives in a soil as. So stem gall of coriander produces what? It is caused by a Protomyces microspores. It will produce chlamydospores that will survive in soil for unfavorable time. So it will produce chlamydospores. Okay. Gloom blotch is a very commonly found in rice, wheat, maize, and barley. So this gloom blotch generally can be observed in maize. Okay. The seed-borne disease, purple blotch of onion, is caused by. So they're simply asking purple blotch of onion is caused by Alternaria pori, which causes a purple blotch of onion. So the answer is Alternaria pori. Expand form of DAPA. So just like expansion. So destructive insect and pest act, which was passed in here India in the year of 1414. So this is the destructive insect and pest act. That is the first quarantine act, we can also say. In India, apple scab was first recorded in the cultivar. In which cultivar the apple scab was first recorded? Ambry. It was the first cultivar which recorded this apple scab. The successful example of transgenic resistance against papaya ring spot virus was obtained using a genetic construct. So I repeat once again, the successful example of a transgenic resistance against Papaya's ring spot virus was obtained using the which genetic construct? Movement protein, replicase protein, and partial protein, coat protein. So this resistance all created by using the coat protein mediated resistance. So coat protein, by using coat protein only, Papaya ring spot virus was managed. Mustard seed like sclerotia are developed in the fungus. So which fungus it will produce? Mustard like fungus. Sclerotia, generally sclerotia produces mustard kind of spores, that is sclerotium, right? So the most susceptible host of a plant viruses are we would say indicator plant for plant viruses, right? Chenopodium amaranthicum, that is generally called as goose foot. Cucumis sativa, Solanum melangina, brinjal, Nicotina benthamina, this kind of tobacco species. Generally, this goose foot, Chenopodium amaranthicum, is the most recommended and the most susceptible host or the indicator plant for plant viruses. Polynitrophenyl phosphate, that is a PNPP, used in ELISA is, so what is the function of polynit? It is used as a substrate generally. So alkaline phosphatase, those are all the enzymes, okay, but this PNPP is used as a substrate. So the next one, genome of banana bunchtop virus. The banana bunchtop virus contains single stranded DNA as a genome, like Gemini kind of viruses, right? Bean anthracnose was first reported by Lindemuth. So that's why bean anthracnose is we used to study Colatotrichum lindemuthianum. How lindemuthianum came from this Lindemuth? The scientist who, or the person who discovered this bean anthracnose is Lindemuth. That's why it's known as the lindemuthianum at the end. So the best indicator plant for potato spindle tuber viroid detection. I repeat, the best indicator plant for potato spindle tuber viroid detection is tomato. So tomato is the best recommended for this identification of this potato spindle tuber viroid. Rust, which is caused by pseudo fungi. So I repeat once again, rust which is caused by pseudo fungi. White rust of crucifers, bean rust, white rust, wheat rust, gram rust. So this bean rust, wheat rust, and gram rust are the true rust, which is caused by Puccinia species, Puccinia kind of species, or other kind of species of rust, like Puccinia, Euromyces. Bean rust is caused by Euromyces, appendiculitis, right? So this white rust of crucifers is known as not a true rust, but a pseudo rust, which is caused by Albuco candida or Albuco bliti, right? So the answer is white rust of crucifers. The fungi which does not cause a powdery mildew. Which of the following fungi does not cause powdery mildew? Okay, Ericife causes powdery mildew. Phylacteria is also powdery mildew genera. Levilla is also powdery mildew genera. Bremia, it is actually a downy mildew genera, which doesn't cause powdery mildew but causes downy mildew. Okay, population of a host in which all individuals have a particular characters of resistance and commonly known as general resistance. In general resistance only, all individuals having the particular kind of characters. Candidatus Liberibacter asiaticus, which causes citrus greening. Citrus greening is caused by Candidatus Liberibacter asiaticus, horn long wing disease we used to call, right? So this is actually phloem limited phytoplasma or bacteria, xylem limited bacteria, xylem limited mycoplasma. This FEB, Fastidious vascular bacteria, which is actually this Candidatus liberibacter, is a phloem limited bacterium. Okay. The time taken to appear the first intact of a virus in a plant cells after the inoculation of 10 hours, very simple. So once the virus is inoculated into the plant, how many hours or how much time after the first virion particles appears on the plant? So after 10 hours. Okay, the virus causing sunflower necrosis disease is a member of ICAR virus. So ICAR virus or carrot rattle mosaic virus or umbra virus, or this comes under ICAR virus, I think so. But sunflower necrosis comes under this ICAR virus only. Who demonstrated first that the pathogen associated with the mosaic disease of tobacco was filterable, which means that can be filtered. So Ivanovsky was the first person who filtered this and told that this it is filterable, so it is not caused by bacteria, because in Chamberlain filter it can filters, I mean it can filters the all bacteria. If filtered by all bacteria, generally this you know one more person, I actually forgot his name, Adolf Meyer. Yeah, I got it now. Adolf Meyer told that it may be caused by bacteria. So after Ivanovsky conducted a Chamberlain filter experiment and filtered all the bacteria. So if it is caused by bacteria, even after filtration also the disease must be caused. Cannot be caused, right? So even after filtration the diseases occur. So it might not be caused by bacteria because the causal organism is smaller than that of bacteria. Resolving power of TEM, transmission electron microscope. So 1 nanometer. Southern blotting is employed for transferring of DNA from gel, so transferring of protein from gel to nitrocellulose, RNA from gel to nitrocellulose, DNA to nitrocellulose, dyes from gel to. You remember only one thing. Northern blotting is employed for RNA. Southern blotting is employed for DNA. Western blotting is employed for protein. Since they have asked southern blotting, go with the DNA. The remaining thing is gel, nitrolux, or whatever, that is not none of our business. This is answer C because it is DNA is involved. So southern blotting is employed for identification of DNA. So transfer of DNA from gel electrophoresis to nitrocellulose membrane. You forgot about all the stuff. You just see the DNA or RNA or protein, whatever it is. We can easily find the answer. Grape fan leaf virus or grape fan leaf is due to the infection of fungi, bacteria, virus, just now I told you, right? It is a grape fan leaf virus, a virus infection which is transmitted by a nematode called Ziphnema index. Onion blast is caused by botrytis squamosa, pyricularia varice, peronospora destructor, magnoporthe griseae. I was a little bit doubted about botrytis squamosa. It is given, but generally blast is caused by pyricularia varice or magnoporthe varice something, but it is mentioned as botrytis squamosa in especially onion. Okay. So the next one is the first systemic account of Indian fungi was published by Bunkur, Butler and Bisbee, Vasudeva Ramakrishnan Subramaniam. So the first account of Indian fungi was published by Butler and Bisbee in 1931 probably. Herbarium cryptogamy, Indian Oriental, so first created by, again this is created by E.J. Butler only. This was we can see in IRA. Okay. First plant virus whose genome is completely genome sequenced. They have mentioned first plant virus. They have not mentioned whether it is a DNA virus or RNA virus. So if it is mentioned first plant pathogenic virus to be completely genome sequenced is cauliflower mosaic virus. If it is RNA virus, it is a tobacco mosaic virus. Cauliflower mosaic virus was sequenced by Frank et al. In 1980. TME was sequenced by Golet et al. In 1982. Dipicolinic acid, which is actually present in endospores, heat resistant acid, we can say. It is an important constituent of bacterial cell wall, bacterial protoplasm, endospore and ascospore. Just now I told you this dipicolinic acid is responsible for the survival during high heat, so which is actually present in endospore. Pollu disease or anthracnose of pepper is caused by it is caused by Coltotrichum gliosporides. Next one is serology. Serologically, which class of immunoglobulins are more important? IgA, IgD, IgG, IgM. Generally, IgG is like the most commonly used immunoglobulin serology. So these are all the answers. For other details and reference, all those things, students can refer this book, A Vision into Plant Pathology, a complete student version. For further doubts and clarification, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.